Welcome everyone. I'm Dr. Patrick Merel, and welcome to this webinar. We have a chance to have during this precision medicine conference Dr. Momo Vuyisich. He is a Chief Scientific Officer at Viome Life Sciences, and I'm delighted to hear with you about his work for the Nutrition 2.0 program they are developing, which, as you can see, is related to data-driven and AI-computed personalized nutrition. And this is perfect for preventive medicine and looking for longer health span. So Momo, I look forward to hear about your work. And please, let's start.
Great. So thank you, and thank you to the organizers. This is a very exciting opportunity for me. So today we're going to talk about data-driven, which means that these are data derived from a person and then AI computed. So computers will actually compute personalized nutrition for each person towards preventive medicine and longer health span. And so when we talk about Nutrition 2.0, just remember this equation, biochemistry plus math. We're going to talk about both biochemistry and math in this presentation and how we can achieve long health spans. Okay, so the concept here of our platform is that we're simply decoding nature. We're not coming up with pharmaceuticals or anything artificial that doesn't exist in nature already. All we're trying to do is decode nature and bring it to every person. And so the concept here is that nature has already solved the problem we're trying to solve, and that is a long health span. So we have currently in the world the living proof of this phenomenon, because there are tens of thousands of people who are in their 90s, and some are even over 100, who live normal, healthy lives. And all we're trying to do is bring that kind of life to everyone, empower everyone to live that kind of a life in a very precise way by measuring their body and informing them exactly what is going on inside of their body, and then telling them what they need to eat and what they need to avoid. So metaphorically speaking, I find this to be best metaphor of our approach, which is that humans have already solved this problem when it comes to air travel. We do not tolerate airplane crashes because they kill a lot of people. And so what do? We understand how airplane systems work. We understand how to do preventive maintenance. And by following specific measurements and preventive maintenance, we can actually keep these planes flying for many, many years without them crashing. And this is, to me, one of the best examples. Airbus A380 is the most complex airplane ever built, and yet in its entire flying history, it has never had a fatal accident. To me, that's the proof that humans are capable of knowing and understanding a complex system, measuring at the right places, at many places, understanding when things are going bad, and then changing them before they actually catastrophically fail. So we just want to apply this same principle to the human body, make a lot of measurements over time, and fix what's broken before it becomes a symptom that's going to display itself as an illness. So what does Nutrition 2.0 look like? It basically goes from a user who provides stool, blood, and saliva samples. These samples are analyzed in our clinical laboratory to generate these biochemical data, which comprise about more than 20,000 data points currently, but this number is going to be growing over time. We then apply mathematical algorithms to these biochemical data to generate these personalized nutritional recommendations. And if followed, they should lead to a long health span. This process has to be repeated every 6 months because our bodies and our microbiome change over time. So some of the highlights of Nutrition 2.0 is that it's 100% data-driven and objective. So it's following two objective sciences. We're measuring molecules using a clinically validated technique, and we're applying mathematical algorithms. So 2 + 2 is always 4. It's never 5, and there is no human that influences this process. So it's 100% objective. It is highly personalized. We have more than 500,000 customers, and we have more than 500,000 diets. Our computers are not aware of any pre-made sort of fad diets like ketogenic or carnivore, Mediterranean, and so on. The goal of this process is to prevent and cure chronic diseases and cancers and to slow down aging. And a very powerful part of our platform is that it's self-learning and continuously improving. So our AI is running 24 hours a day, 7 days a week to extract as much data from all of the data we've collected so far in order to improve these mathematical algorithms and make them better and better.
Momo, can I interrupt you to make this conversation maybe a little bit interactive, and as well not too afraid our attendees, specifically I'm thinking to our clinical colleagues. When you talk about collecting 20,000 data points, I don't want to afraid everyone here about massive testing options, but without too much spoiling the rest of your presentation, can you rapidly let us know about what do you have in mind when you mentioned collecting 20,000 data points, because this is a huge amount of data here.
Yeah, very good point. So first of all, we only perform 3 tests. So this is not something that we split blood into many, many tests. We only need 50 microliters of blood. We only need 100 milligrams of stool, and we only need about 100 microliters of saliva. And so only 3 tests are performed, but these are next generation sequencing, very high throughput tests that quantify RNA molecules. And we'll get into RNA versus DNA later on. But these 20,000 data points can be grouped into 3 categories. One category is the relative abundance of every single microbial species in all samples, the microbial gene expression, so all of the microbial functions are quantified, and then human gene expression, or all of human biochemical functions in blood are quantified. And so those data combined, which we will break down a little bit further, make up more than 20,000 data points.
Great.
Okay, great. So what does this look like for a user? A user will receive a kit at home. All samples are collected at home, including the finger prick blood sample. The samples are actually shipped at room temperature across the world because they're stabilized with our special preservative. The samples arrive in the lab, and we use a fully automated, CLIA certified, this is a clinical standard in the United States, laboratory to obtain these 20,000 data points. Mathematical algorithms are then applied to these data, and the user sees the output as results and recommendations. The results consist of microbial species identified and then a bunch of health scores that refer to different aspects of the human physiology, and then very detailed recommendations of all foods in terms of the amount you should be consuming them. So they go from superfoods to enjoy foods, to minimize foods, to avoid foods. And then also every user receives very, very personalized and highly detailed supplement recommendations as to what supplements they're supposed to be taking in what amount. And so all of that is listed on the app or the web account, and then the person is supposed to follow those recommendations and then repeat this process every 6 months. So that's the future of us being treated as airliners, where every 6 months we get a full digital profile of our body. We know exactly what works well, what doesn't work well, and those things that don't work well, you're going to be informed exactly what you can do about it, so that 6 months later, hopefully you will see an improvement. Okay, so now let's talk a little bit about a very important topic, which is root causes of chronic diseases and cancers, because I think there's quite a bit of conflicting information here. So I want to clarify that. So the statement I want to make up front is that most chronic diseases and cancers are not either bad luck or a genetic fate. They are simply a result of biochemistry that we have not yet figured out. And so we just need to figure out that biochemistry, and then we will identify root causes of chronic diseases. Now, I'm making that statement based on the vast amount of research that has been published over the last decade backing up that statement. And I'm showing here 3 papers, but there are many others. And these studies have been done in very large populations, and they have been done also in genetically identical twins, where the genetic background is the same. And I cannot go over all of the highlighted areas, but you can pause the webinar and then look through all the highlights. But basically, the summary here is that genetics makes a minor contribution to chronic diseases, and on average, it's in the single digits in terms of the percentage of importance. And so when when you're looking at your genetic data, or when you're thinking, well, I have some disease running in my family, or when you say, I don't have any disease running in my family, all of those mean that you should still follow the best nutritional and lifestyle recommendations that you can, because you can actually determine your own fate independent of your genetics. Another concept that I would like to clarify is that we as humans are mostly not humans. And let me explain what that means. So if we look at the human body, what we see and what we used to see without a microscope was just human tissues. But it turns out that microbes are more important inside and on the outside of our body than our own body. And so just some numbers that I'm putting out here are that humans encode 23,000 genes. So we are capable of essentially performing 23,000 different functions. Whereas the microbes that are found inside and on us, they have the capability to produce 10 million different functions. In reality, while most of the human genes are expressed, a very small percentage of these genes are expressed, but still the ratio is at least 1 to 10, which means that we're still very little human. We're a small part of our ecosystem, and we're mostly microbial. And so the reason why genetics has failed its promise back from the 90s when we sequenced the first human genome is that human genetics plays a minor role in chronic diseases and cancers, which is why many chronic diseases and cancers have actually increased in prevalence over time, because genetics hasn't been able to solve that problem. So we have to look not just at genetics, but we have to combine it with epigenetics. And that's sort of the focus of this talk, is epigenetics. I also want to be sure that the audience understands the power and the importance of microbiome with a very sort of eye-opening example. So in this particular example, a woman was sick from bipolar disorder, and she had tried all pharmaceutical approaches, and she was still sick. She couldn't function. She couldn't hold a job. She didn't have a good relationship. She didn't have a good life. And all she did was she went to a doctor and conducted a fecal microbiota transplant. So she took stool from her husband and transferred it into herself for a few months. And that process completely cured her of bipolar disorder. And this has actually been documented in this peer-reviewed paper, but actually another peer-reviewed paper came out also showing that bipolar disorder can be cured with fecal microbiota transplant. And this kind of work has been done in many animal studies where people have been able to transfer the illness from the human to an animal just by transplanting gut bacteria. And so this is a very powerful demonstration that maybe the concepts that we were convinced of, such as that bipolar disorder is a disorder of the brain, may actually not be true, that we need to look at the systems biology view of a disease, and that very likely bipolar disorder is the result of both nutritional and microbiome factors more so than our own genetics. Over the last decade, microbiome research has also shown very strong associations between gut microbiome and oral microbiome and the vast majority of human diseases. So this is now a very well-known and accepted fact that human microbiome influences the onset and progression of cancers. And I'm not going to go into details here, but I can also mention that gut microbiome can actually, in a causative fashion, determine the outcome of immunotherapy for cancer. So I encourage the audience to go and research this area of the influence of gut microbiome on whatever disease they're interested in, including cancers and response to immunotherapy, and how a simple change of the gut microbiome can reverse a non-responder to a responder to immunotherapy, for example. Okay, now we get into some hardcore biochemistry. And so this is very exciting, very important, but it's also a little bit technical. But I will make it as simple as possible. Here we're going to go from a specific microingredient in foods, a specific type of a fiber or prebiotic, through processing of that fiber by gut microbiome into a metabolite or a chemical called butyrate. And then that chemical that's produced by the gut microbiome translocating over into the bloodstream and influencing the fate of our immune system and how we respond to an infectious disease. So let's dive deeper into this. We go from a specific fiber or prebiotic that ends up in the colon because we are unable to digest it. Here we have specific bacteria that can process that fiber into a compound called butyrate. So butyrate is a small molecule, and it's a byproduct of bacterial fermentation of these fibers. And when that butyrate goes into the bloodstream, it signals, it's a chemical messenger that tells human T cells, which are involved in response to viral infections, it tells the T cells to convert themselves to memory T cells. And this becomes very important. And we now have a very relevant case of COVID, for example, where some people get COVID once or they get vaccinated once and they don't get sick again. But some other people, whether they get COVID or get vaccinated, they get repeatedly infected. And the question is, why is that? And this is one of the reasons. There are other reasons. This is not the only one, but this is a major reason. And that is that as CD8 positive T cells are responding to a viral infection, they can clear the virus that time, but they have to convert themselves to a memory T cell in order to stay in the bloodstream and be ready to recognize the next infection. If they don't convert themselves to a memory T cell, they will die, and your body will lose the memory of that infection, meaning that next time you encounter the same virus, you may get sick. And so this is a chemical messenger produced by the gut microbiome in the colon in response to a specific micronutrient. And when that specific micronutrient is not introduced in the body, then that specific butyrate or chemical messenger is not produced, which means that human T cells will not become memory cells, and you increase the chances that you're going to get reinfected by COVID or any other virus. This is a very nice and elegant mechanism that has been determined and published, showing how specific micronutrients can influence our own physiology in a very profound way via the gut microbiome. And all of these measurements here, all of these reactions, chemical reactions here, we can measure via RNA, but we cannot measure via DNA. And we're going to go into that. But we can actually measure the activity of this pathway that goes from fiber to butyrate, and this pathway that goes from human T cell to memory T cell using RNA or gene expression analysis, whereas with DNA analysis, you cannot actually measure this process.
So tell me, Momo, at this stage, you are meaning that this is not only the presence of specific nutrient in our diet or a specific microbiome in our gut, but this is a combination of two.
Yes.
So which that indeed butyrate is not produced by our body. So we need to bring it to facilitate these T cells to memory cells conversion, right?
Great point. Exactly. Butyrate cannot be produced. We do not have the genes to produce butyrate. So butyrate can only be produced in the colon under anaerobic conditions, so no oxygen condition in the colon. Yes. And so this is a true example of a synergy where we have co-evolved with the microbiome. It's not that they're a parasite or bad for us. They're actually incredibly important for our physiology. We have integrated their biochemical activities into our body so that we work together. And it's dependent on food. And while we're here, I want to mention that these fibers, one of the reasons why I mentioned this specific example is that as cultures shift from sort of not developed to developed, one of the nutritional changes that occurs in a society is that we go from more raw, untouched foods to more highly processed foods. And this is one exact example where people used to grind their own wheat and make their own whole wheat bread. But as they move to the cities and become more financially independent, they like to buy white bread and they like to buy white pastries. But what happened in the process is that the food industry has removed these fibers. And so when you're consuming a white bread, you're not actually benefiting from these fibers. Whereas if you go to whole grains, you're actually benefiting it. Now, one of the major problems in the grocery store these days is that the food industry will label a bread something like whole grain bread. But if you look at the nutritional ingredients, the main ingredient is just white processed wheat. And then down at the very bottom, they say, whole grain. But what they do is they actually make a white bread and then they sprinkle some seeds on top and they call it whole grain bread. And you believe that you're eating whole grains, but you're actually not. And so you have been tricked into consuming foods that lack fiber, and this is just one of many mechanisms by which fibers feed our microbiome, and they actually make us healthier. There are many other mechanisms. One that I would like the audience to research is how fibers that are present in human breast milk actually feed the microbiome of the baby in order to make that baby resilient and develop a healthy immune system. And that all goes through the microbiome. But we don't have time to expand on this topic because there are many other examples. Let's continue with the biochemistry. So I promised you that we would discuss the biochemistry component and the math component of Nutrition 2.0. And the biochemistry relates here specifically to measurement of RNA. And so let's talk about why RNA. Most people are familiar with DNA and have heard DNA sequencing, but most people are not familiar with RNA, and that's why I have to introduce this concept. So every person in the world has about 23,000 genes in their genome that they inherit from their parents. And these genes encode different functions, but a gene itself is just a physical object that doesn't actually perform any function. It's just a blueprint that sits in space, and if expressed or activated, then it will perform its function. But until it's expressed, it's just a physical object that doesn't do anything. And you can see here a dramatic example of that, where if we sequence the kidney, liver, and brain of one person, and we sequence the DNA, we will find that all of these three organs have the exact same DNA. Every cell in our body has the same DNA. And when you sequence that DNA, you're going to find that, oh, wow, it has 23,000 genes, and those genes have a potential to be either brain, liver, or kidney. But I don't know which sample it came from because it's just a potential. But if we sequence the RNA from kidney, liver, and brain, meaning which genes are activated and to what degree they're activated, that's called a gene expression profile, they're going to be vastly different. So brain cells are going to only activate a small subset of all the genes that enable brain to perform its own function. Liver cells will activate a different subset of genes that enable it to perform its own functions, and kidney will do the same. So what makes these tissues what they are is not their DNA, it's their RNA or gene expression. And that's one of the reasons we measure gene expression and not the genes. Now let's go to a more dramatic example where we now can distinguish health from disease using RNA, where we cannot distinguish it via DNA. So this is a single patient who has been diagnosed with Crohn's disease. This is his intestine, small intestine, during the time of remission, and this is his small intestine during the time of relapse. And what you can see here is very, very different morphology. Here he's asymptomatic, and here he's in pain 24 hours a day, and he cannot eat. He has 15 diarrheas per day. If you sequence DNA of these two tissues, because it's the same tissue, you're going to get the exact same result. And that DNA cannot possibly distinguish between health and disease because it's the same DNA. But if you measure RNA in these two tissues a week apart when they're in relapse versus remission, you will see a very, very different gene expression profile. This tissue is expressing inflammatory genes that make this tissue look and feel the way it is, whereas this healthy tissue is not expressing those inflammatory genes. And those genes can all be seen as being activated. And this has all been published. And this is another very important reason why we measure RNA. Another example that I can bring up in the microbial world is that a microbe has a potential to express genes that produce healthy biochemicals for the host, for us, and has the potential to produce harmful chemicals. And which ones it's producing, the only way you can tell is if you look at the gene expression. You cannot actually sequence the DNA of that microorganism and know what they're doing at the moment. So we decided early on to go for RNA sequencing instead of DNA sequencing. Okay, so now let's go to what does this physically look like? And we're going to now dive deeper into these 20,000 data points. So this is an actual sample collection set that our customer just sent in yesterday. So this is the stool sample, saliva sample, and 2 tubes of blood. This is 100 microliters of blood plus preservatives. So there's only a a total of 200 microliters of blood here. These are all preserved so that they can be shipped globally at room temperature to preserve RNA. And so when we analyze these three sample types, as we discussed earlier, we get more than 20,000 relevant data points, and they consist of relative activity of microbial species in the colon and saliva, which are these numbers, the expression level of genes or KEGG orthologs. This gets a little more technical, but think of KOs as biochemical functions, for example, to produce that butyrate. We quantify about 4,000 active functions, microbial functions in the saliva and in the stool. And then we quantify the expression level of about 12,000 human genes on average. So all this adds up to more than 20,000 data points. And this is basically where the physical world ends. We quantify these in our laboratory, upload these data into the cloud, and now the mathematics takes over. Just to be a little more clearer, for example, if we look at these 1,250 microbial species and their relative abundance in the colon, what does that actually look like? Well, if we index the species here on the x-axis, so this is 1,500 species, there's a name to every single one of them, which I could not display here. You can see that their relative abundance in percentages ranges from about 10% all the way down to like parts per million. And so every species has a certain relative abundance, and they all have biochemical functions that influence our health and disease. One more very, very important aspect of RNA that DNA does not have is that RNA can be modulated. So just like I showed you the example of a Crohn's patient where one day they're in remission and their RNA is having a certain gene expression profile, but a week later they go into relapse and their RNA profile changes, but then a month later it goes back to remission. That tells us that RNA can be modulated. And how is it modulated? It's modulated via chemical messengers. And these chemical messengers are either directly introduced by foods, or they're actually byproducts of microbial digestion of the foods. And so, in other words, we can actually modulate all of these biochemical activities using nutritional ingredients. And here I'm just showing you just a slide that I will go deeper into when we go to the live session in Dubai. But in this slide, we're showing these molecular scores where we quantify specific microbial functions in the gut and the saliva. And you can see here in blue are the initial scores for our users, and in red are their scores after they've been following our nutrition for at least 3 months. And you can see here that 15 out of 17 scores have improved in our user population, and 10 out of 12 oral scores have improved in our user population after 3 months, meaning that nutrition can actually make a real positive impact on the biochemical functions of the microbiome that have been associated with human health and disease. And so this is really a very dramatic advantage of RNA over DNA in that you can actually do something about it in a very short period of time. Any questions, or should we switch to math?
No, it makes me think. I may have 2 questions for you at this stage. I hear you between the difference between testing the DNA versus testing the RNA. I agree that different tissues will have different gene expression, but at the end of the day, this is coming from the DNA, and this is probably importance of epigenomics. So did you explore this way as well? Because now with sequencing technology, you can produce whole genome sequencing at the same time with the epigenomics. So that might be an interesting way as well to explore this relationship with our microbiome.
Yeah, so that's a good question, and we did explore epigenetics, but to me, ultimately, the actual phenotype of a tissue is going to be determined by the gene expression. Epigenetics does influence gene expression, but it doesn't always influence it in the same way. Depending on where the epigenetic loci are, they can either reduce or increase the gene expression, and ultimately, any measurement of epigenetic markers are going to allow you to only build a model of what gene expression looks like, whereas we are actually measuring very quantitatively the actual gene expression. So we do not need the epigenetic data from DNA in order to model anything because we measure it. Does that make sense?
It makes sense. Yes, absolutely. But in that kind of consideration, why not taking as well the metabolism analysis?
Yeah.
Because indeed, you showed beautifully our nutrition can bring some molecules that are necessary to facilitate our immune response, for example. But we eat every day, so we bring everyday metabolites, and maybe the level of metabolites that are brought during our regular diets has its importance as well. So how can you connect to, let's say, your patients, your participant regular diet and the level of metabolites they already have on board, I should say.
Yeah. Yeah, that's a very good question, and we would love to do metabolomics. We would love to do it. But there are some fundamental problems with metabolomics. One is technical and one is physiological. So let me explain. On the physiological side, if you look at that butyrate, when that butyrate is produced in the lumen of the colon, what's going to happen is that what we didn't discuss, but that butyrate is actually acting as food, as a carbon source for the intestinal epithelium. So some of it is going to be absorbed into the intestinal epithelium and consumed. Some of it will be excreted into stool, and some of it will be translocated into the bloodstream. So if we want to really follow and measure that butyrate, we're going to have to measure it in both stool and blood. And that applies to many others so that you know what's happening to it. Technologically, metabolomics, unfortunately, has not reached the level of consumer friendliness that metatranscriptomics has. So we spent 6 years, I spent 6 years with my teams prior to starting my company, working out metatranscriptomics in order to get it to this level where we can ship kits all over the world, we can collect the samples and ship them at room temperature over to our labs. That kind of a mechanism has not been developed for metabolomics. Sample collection and preservation for metabolomic analyses are much more complicated. And once a sample arrives, let's say you collect a stool sample to do a metabolomic analysis, you're going to need to, by current methods, split it into 4 samples and perform 4 separate metabolomic analyses. And today, that can be reduced down to about $1,000 per analysis. But if you're going to then add blood, that's another $1,000. And so you have to take all these practical considerations into account. And to me, metabolomics is simply not ready for this kind of scale and affordability that metatranscriptomics is. Could it add value to our platform? Absolutely. And we are a systems biology company, and we will add any other omic method whenever it becomes valuable enough, meaning that it has to be providing overall value, meaning cost versus the benefit, right? So yeah, good discussion.
So to excite you to come to the conference as well, our auditors, I believe that we will have a presentation about recent development in metabolomics technology and AI that I look forward to hear about the cost and the practicability, because indeed, bringing transcriptomics, microbiome, metagenomics testing, bringing this kind of testing solutions at a large scale. Indeed, you mentioned the cost of analysis. It's a big point. So I believe that you have worked as well trying to reduce the cost as much as possible of this combined omics technology.
Yes. Yeah. I mean, we have $1 million worth of LC mass spec equipment sitting in my lab, and we spent 2 years trying to work this out, and we just couldn't. And so we gave up on it for now, and we're simply waiting for someone else to invent this technology so we can adopt it. So hopefully others will.
Okay, look forward to the conference. So
Great.
see we have new technologies specifically in that space.
That would be great, yes. Okay, now let's talk about the math. So the other part of Nutrition 2.0 is the math. So how do we interpret all these biochemical data using mathematical formulas, and how do we translate them into personalized recommendations? And so here's a takeaway slide. So this is one of the slides that you should really take away, because it sort of explains how you go from all the way from the beginning to the end in terms of machine learning. So what we do is we collect data from all of our customers and from all of our research participants. So we have more than 500,000 customers in more than 100 countries, and we have more than 16,000 clinical research participants. So we collect those molecular data that we already discussed, the biochemistry, but we also collect very comprehensive metadata. We collect clinically validated surveys for 36 different diseases. We sometimes collect medical records from our clinical research participants, and we do daily, weekly updates on their diet and how they're doing. And so we get massive metadata combined with these 20,000 molecular data points. We load all that up into our data lake. And so we're working already today at petascale computing, and AI can then tease apart what are the important molecular and phenotype data or metadata for different diseases. And so this is what a machine learned algorithm would look like, or machine learned model would look like. This is a model. It would basically, machines would come up with what are the features that you measured. So this can be like the relative expression level of a human gene. This can be the relative expression of a specific microbial gene. What are the features that are strongly predictive of that disease? And what are the features that are anti-correlated with that disease? And the computers will actually quantify the relative contribution of every one of these features. And so this is the biomarker set for every disease. And we use these biomarker sets for multiple purposes. And so we use them for both making nutritional recommendations and for building diagnostic models. So let's dive a little bit into one specific example. So type 2 diabetes and high glycemic load in the bloodstream has not only been associated with prediabetes and diabetes, but also Alzheimer's disease, heart disease, obesity, and so on. And so reducing the postprandial glucose response, or the post-feeding blood sugar, is a very important medically relevant technique. However, if we look at the glycemic index that's available out there, this is a glycemic index for an average human. And this is unfortunately very misleading, and I will show you why this average glycemic index is misleading. Here are actual measurements from a continuous glucose monitor for 2 people that consume either a slice of sprouted grain bread or a banana. And you can see here that one person, when fed a banana, their sugar spikes from about 80 to about 160, but the other one stays relatively flat. And when this person consumes sprouted grain bread, their sugar doesn't jump very high, whereas this one jumps much higher. And so you can see here that the glycemic index is highly personalized, and that we have to inform people what they're sensitive to and what they're not sensitive to, so they can adjust their diet and not follow this average recommendation for an average human being. And so for that reason, we conducted a very large, very complex study. We recruited 1,100 participants, half of them in the USA and half of them in Japan. We went for USA and Japan because these 2 cultures have very different genetic makeup, very different environments, very different nutrition, foods, food chain, and so on. And so we wanted to develop something that we want to first see how universal it would be, and can we make it universal? We collected their stool, or in some cases, stool, blood, and saliva at the start. And then for the next 14 days, we mounted a continuous glucose monitor on each participant, on all 1,100 of them. And during those 14 days, they collectively consumed 60,000 meals. And these 60,000 meals were very carefully designed to include 104 different kinds of meals that represent different ratios of carbs, fats, and proteins. And we recorded the glycemic response to every single one of the meals. And then we used machine learning and asked the question, could we have predicted this kind of a response for every single person just based on that stool sample, without actually going through different diets and monitoring with the CGM? And the answer was yes. The machines came back and they learned how to do that. And they learned that very effectively. And so we published an article on that describing all the methods, but basically every single Viome customer benefits from this machine learned model in that it's a part of the recommendation engine that takes into account what is going to be the glycemic response to that food for every customer. While we're here in the type 2 diabetes area, we also use machine learning on a very large data set of over 50,000 people to learn what are the molecular features in the gut microbiome that are very strongly correlated with health and then prediabetes and then diabetes. And we developed that model that's very, very statistically significant. And now we can use these biomarkers, just like what I showed you in the first slide, for Nutrition 2.0 algorithms, but also as a molecular endpoint for clinical trials. And speaking of clinical trials, in my live talk in Dubai, I will present several the results of several of our clinical trials and how this Nutrition 2.0, when implemented in real world, in direct-to-participant clinical randomized controlled trials, what the efficacy is. So I'm really excited to present more data and continue this discussion live.
Beautiful. We can't wait. What I would like to react on your before last slide, when you succeeded into designing machine learning algorithms to predict diet adjustment, because this is what we are talking about. With this type of program where patients will come every 6 months, as you mentioned, what about leveraging wearables that are coming today on the market? And you mentioned your study with providing glucose monitoring to participants. Do you think that this type of wearables will be beneficial to have to adjust your algorithms probably in real time at some point?
Yeah, so we can be adding many pieces to our platform. We are now starting to add, for example, activity monitors, so we can actually get health data from Apple Health. We can certainly be adding CGM data as well. We can be adding any other data, heart rate variability and heart rate and blood pressure and all that. We can, but our goal really is to make this as simple as possible and not make it complicated. So our goal is, can we see biochemical signatures that are very detailed and that are actionable? And if we can, then we can use the exact same 3 tests and the molecular data derived from those 3 tests to inform the person, because in our experience, people are busy and people don't like to read instructions and don't like to do things and extra things. It's a generalization. It doesn't apply to everyone. There are certainly people like me who I like to put a CGM on me all the time, no problem. Sometimes I'll wear 2 of them, right? And early on before the study, I did that all the time. But still, at some point in time, life gets complicated and you don't want to be adding too many things. So what we're looking for is, how can we simplify people's lives, first of all? And second, if we see good molecular biomarkers of something, then there's no need to add additional things. But for example, for physical activity, it'll likely be necessary to collect physical activity data in order to improve our algorithms. So we are looking forward to that.
We know how human being is. People want results rapidly. They would like to have some kind of incentive to do this. So results that are reported, let's say, almost real time, I'm pretty sure that there will be a big interest in the communities. So indeed, glucose monitoring devices, it's kind of additional burden. But between the choice of returning every 6 months for a full analysis, well, we can ask the audience during the conference, because I believe there will be a lot of wearable companies involved in this kind of studies. And I look forward to hear what they have to say in that space and how useful they can be to the kind of work that you are doing, specifically related to nutrition, diet, and life adjustments, because people, if we want to get their attention, we need to provide not only information, but tools to achieve their goals, probably.
Agreed. And I'd like to say that a CGM standalone is probably going to provide a little bit better data than what we can provide. However, please remember that the prediction of whether a food is going to have a high glycemic load or not for each person is only a part of the algorithm. So we're not just looking at right. We're not looking at just reducing the postprandial glucose response, but we're looking at thousands of different biochemical activities, and all this has to be taken account. And our computer will often make a decision that even if something is going to spike your glycemic response, if that food contains a very necessary ingredient, and in marginal cases, if you have reduced your microbiome diversity to the point where you only have 1 or 2 microbes that are capable of producing some biochemical that's very good for you, and they require specific micronutrients to perform that function, then the computer will put a lot of weight on that and will actually reverse the glycemic response, because it'll say, well, I have to make a choice. You're either going to have some very important deficiency, or you're going to have a high glycemic response. And the computer, because the computer has a weight for everything, it can make a decision to reverse that. Whereas if you're just reading your CGM, you would not have that information. So I think this is what we're doing is a nice comprehensive approach. But if you want specifically to focus on one phenotype and only one phenotype, I do think that CGMs would provide a slightly better accuracy.
Beautiful. I look forward to the rest of your presentation.
Yeah, thank you.
Look forward to have you all in Dubai for precision medicine.
Yeah, I'll see everyone in 10 days or so. Thank you so much, Patrick.
Thank you, Momo.